So thank you. Thank you, Christina. And then I would like to welcome Sam Baxter for a talk about prevalence on 4-H. But I think, as I said in my invitation, I think this is important for other rare disorders too. Sam is a genetic counselor, a licensed genetic counselor, but she's also as an associate director at the Broad Institute, Broad of MIT and Harvard, where she heads the group that is responsible for genetic and genomic data sharing. And as I said, this collaboration came together through our work at CZI, and we are very, very happy to have her. And I'll just hand over to Sam. Without further ado, welcome to everybody who joined us. I'm glad we have new people, and I'm glad that others have joined again. So let's go ahead. Thank you.
Great. Thank you, Valerie. So as Valerie mentioned, I'm a genetic counselor, and I always lead with that because I think that my lens and my take on all of this research that I do really is colored by the fact that I'm a genetic counselor. You'll also notice as I speak, I'm a very informal speaker and love to have questions throughout. We do have, 1 hour to cover a lot. Typically when I return these results to groups, I book an hour and a half and we fill the time quite easily. So I will try to kind of keep things moving, but I can stay on later. So if anybody wants to stay on past the hour and ask questions, or if there's additional things that people want me to dive into, I'm happy to stay on and go deeper into that information. So let me just quickly make sure I can share, or we know I can share my screen, but go ahead and share. While I'm doing that, oh, hold on. All right, can you all see my screen appropriately? Yes. Great, wonderful. So one other thing that I wanted to add for my introduction, just because I think it's helpful to understand the context of where I'm coming from and what I can answer questions on. I'm also the operations manager for gnomAD, which is the database that we use for the allele frequencies for these calculations. And so if there's any questions about gnomAD, what is present in gnomAD or is not, please feel free to ask questions about that as well, because I think that's helpful context. And I will talk about gnomAD throughout this as just a point of reference for people to understand what are the pros and cons to using population databases in this context. So the title of the talk is Polar 3 related leukodystrophy, not so rare, but this really is a larger study, prevalence study that is funded by the Chan Zuckerberg Initiative and is part of the Rare Genomes Project at the Broad Institute. And we do this with quite a few patient advocacy groups. And so I'm gonna really give a high level understanding of what the prevalence study is, but anchor it with Polar 3 and hopefully be able to show you why this is not as rare of a disease as once thought. I will also be talking a lot about the work that we do. It's really the royal we. I have a very large team of people that work with me, specifically some of my variant curators who are actually on the call today, including Moriel Singer-Burke and Katie Russell, who do all of these variant curations that you'll hear me talk about, which is a lot of work, and I want to make sure that they get absolute due credit for this. But I also have Anne O'Donnell-Luria and Heidi Ream, who are the PIs that have helped guide me throughout the work that we've been doing. All right. So this is like I said, the genetic prevalence study, which is really, to me, a puzzle of putting the pieces together. So all of this information is data that lives in the open source space. It's all really anchored and driven by data sharing and databases where the community has collaborated together. But it takes the opportunity of putting all this information together to really weave the story about one of these diseases and genes. And there's nuance and differences for each of them. And I'm going to talk about some of the high-level themes that we've learned as we've done this across a lot of different genes. So really the point of this study is to answer the question, how many of us are there? And the us is in question marks because that the definition of us changes with each group that we work with. But on the left-hand side, you'll see 0. So this is in the case where there is no patients with the disease, there's no gene implicated. And we know that there have been studies and ways and methods of figuring out how many of us are there that's been anchored around clinical cases. So your traditional definition of incidence and prevalence. This is often defined as individuals with either a molecular or clinical diagnosis of a disease, is often gathered through public health efforts, through collecting medical records, literature, genetic testing labs will contribute to this, patient registries when they are available. But we know that this is traditionally underpowered and are lower numbers. So it's very rare that we have a patient advocacy group that comes to us and says, we have these numbers and we think this is completely accurate and we've captured everybody. Typically, it's we know we're missing people. But the question is, who are we missing and where are they? So what has emerged now is more population databases analysis, where it's anybody with, or any individuals with 2 known pathogenic variants in a recessive gene. And this is traditionally what has been sort of assessed as genetic prevalence. But this is often based off of annotation only, meaning they say, go into ClinVar and pull anybody who says that they have a likely pathogenic or pathogenic variant, or go into X database, and we just want to take it at face value. There's absolutely a use for this, and I'm in no way saying that this isn't a great place to start. It gives us a sense of who may be carriers in the population, who may be at risk to have a child with a certain disease. But there's a lot of limitations when you take things at annotation only. We know that for a long time within ClinVar or other databases like HGMD, which are all databases we use and I'll talk about more, that early days, it was a lot easier to call things pathogenic and likely pathogenic, but that didn't necessarily mean that it was true. And a lot of that historical data still resides in these databases today. And so we need to take sort of today's knowledge and apply that to variants that have been seen in the past. So the result of that is that we think that these are a little overinflated numbers as we dive in a little bit more. And so I've indicated that by showing a lot of people, but only a few of them have these double helix, saying that they truly have both the clinical and the molecular diagnosis. So what resides somewhere in the middle is the true genetic prevalence, but where that resides in this spectrum is something that we're still trying to zero in on. And what I define as true genetic prevalence is the estimated proportion of a population that either has 2 compound heterozygous or 1 homozygous disease-causing variant. The reason I'm not saying pathogenic, likely pathogenic, is because we've sort of opened up the spectrum a little bit. Again, we're talking about an estimate. It's going to be very hard to say this is the exact number because these databases are always changing. But that is what we are trying to zone in on. Who are the individuals that have either 2 compound heterozygous or 1 homozygous disease-causing variant? And we use both clinical curation database and open source databases, including population databases, to get there. Now, when we look at this spectrum, there's a few places that we can fall on that when we perform this analysis. So if you look at 1, which is we would get a number that is smaller than the number of clinical cases that have even been reported out there. This is not a result we want to get, but we have gotten that before. What that usually is showing to you is that the population that is most heavily impacted by a disease, or maybe have a higher carrier frequency, is not represented in the population databases that we are looking at. And I'll go into that a little bit, and you'll hear me harp on over and over again. One of the key things that we need to think about when we're talking about this type of analysis is who is present in our population database, in this case would be Nomad, and who is not present in the database. And certainly seeing that we're seeing numbers much lower than what even clinical studies are showing is a sign that we are not capturing that population in the database, and you need to think about that when you're looking at the results. Position 2 and 3 are sort of we know that we're getting closer to the true genetic prevalence, but we might still be undercounting and we might still be overcounting the number of people. And that changes based off of every gene and every disease and what we're looking at. And there's sort of clues and hints that you can look at by talking with patient organizations, looking at the literature to understand where you may think you'll be. And so we'll come back to this at the end to talk about where I think we are with POLAR 3 at this point. So how do we build this puzzle? I keep talking about this method. Well, what is this method? Well, if we're going to build this puzzle, we really have 5 key steps that we need to start with. The first is our sources, the places that we're going to pull information to start. If we're going to estimate the prevalence of a certain disease, we need to start with what are the variants that are leading to the disease and what are the genes that are implicated with this disease. And so we start there. After we've come up with that, with the sources that we're going to pull this information from, then we go to curation. And curation is the process of looking at the genes and looking at the variants and deciding how well we think the evidence has stacked up to call something pathogenic or disease causing. Then we hit in the middle where we say, okay, we have our final list of variants, but even those have a little bit of gradation. They have some color to them. There's not everything is established pathogenic. Sometimes there's likely pathogenic. Sometimes there's variants of uncertain significance, but it's early on and we really think there's some information there that might lead one way or another. And we found that there's a huge power in talking about this in ranges and in, it could be here, it could be there, and here's the reason why. And so it's critical to also annotate the classification and the sort of the heaviness with which you're weighting something. We then move on to aggregating the frequency of those variants using the population data. And again, going back to understanding the nuances of what is in a population data and what's not and how we think about that. And finally, the last step, we move on to the prevalence calculations, and we put that in the context of the information that we know about the disease. So where do we start? Well, if you're anything like me, when you got a puzzle in front of you, you first sit down and you find your corner pieces. And that's really the sources of information to me when I think about this puzzle. The absolute critical thing to start with is where are we going to pull this information from? So for this particular study, we started with 4 key databases, HGMD, which is a database of variation that really is pulled from the literature. So we take any variant that's been designated as DM, which stands for disease mutation. We know that this is historically over-inclusive. There are a lot of variants that if you put them through the full curation, they don't make it to the full pathogenic, but we want to be as inclusive as possible. So if anybody has ever said they think it's pathogenic, we're going to review it. Next, we go to ClinVar. This is a database that is used heavily within the clinical genetics community and in the research genetics community, where laboratories, both clinical and research, and also even patient organizations through a system called Genome Connect, can contribute data to this database and say, we've seen this variant. Here are the people that we've seen it in, not actual individuals, but giving high-level phenotypes. And here's the classification that we gave it. They can even provide the evidence. That's not always there as often as we want, but there is sort of a backing of the evidence. And from that database, we pull all variants that have been annotated as pathogenic, likely pathogenic, or where there's a conflicting classification where one of the conflicts has either been pathogenic or likely pathogenic. We also work with all of our patient organizations, their network of clinicians and researchers to see if they have any disease-specific databases that they rely on or would like to use. And we pull in really for those groups, anything that they give to us where they've had suspicion of pathogenicity. These are the people that know the disease best. And so we want to take them at their word, but we do put it through the full curation. We are happy to say that most of the time, these variants have either been listed in ClinVar or in HGMD. And so we're not often missing some when we can't get that information, but we like to make sure that we're including as much as possible. Finally, we go to Nomad and we pull 2 different pieces of information from Nomad. The first is we pull all the high confidence loss of function variants. The other databases I've talked about certainly have loss of function variants. These are variants that are thought to actually lead to nonsense mediated decay, meaning that the protein is not there or is absent. But this is in Nomad, what we're looking for is not whether those have been reported in individuals. We want all of the variants that have ever been reported at or have are called loss of function by the annotation. And we have an algorithm called Lofty that assesses to make sure it's not in the last exon of the gene or in certain areas of the gene that we are questioning whether it really would lead to loss of function. And that's what the designation high confidence is. But algorithms are algorithms. There's blind spots to it. There's things that a human being can look at and understand better and kind of can make a more specific call. And so we do apply another level of curation that I'll talk about. But the benefit of bringing in these loss of function variants is that we know not everybody with a disease has been sequenced. And so certainly there are populations that have even less available sequencing than we have in the US or in Europe. And so we want to make sure we're saying, if we know that loss of function is the mechanism for a disease, which it almost always is for recessive disease, that we're capturing as many of these variants as possible. The second point of information that we bring in is the allele frequencies for the variant. So how often the variant was seen in certain populations. We filter all of these databases on the allele frequencies. The reason for doing that is if the variant is not present in NOMAD, it's not saying that it's not pathogenic, but we're not going to put it through the full curation, because when we put it into the final calculations, if the allele frequency is 0, it doesn't change the prevalence. And I absolutely adore my variant curators, and I don't want them to have to curate variants that are not going to change the numbers at the end. And so we do this as a way to triage the amount of work that is put onto the variant curation team. But absolutely, one of the limitations of doing this is that there are pathogenic variants out there that are not present in this population database, and those aren't being captured in this. The thought is that overall that they are more rare, but they could be more common again in populations that are not represented in NOMAD. And so just keeping that at the forefront of your mind as you're thinking about these calculations is incredibly important. So I keep talking about curation. I think many of you probably do know what curation is, but for those who don't, curation is the process of looking at the evidence around a variant. And there's a lot of different types of evidence that we look at, including like I've talked about population data, but also computational and prediction models that say if you change from one amino acid to the next, do you think it disrupts the protein versus not? Functional, so actually, in the lab, looking at different types of studies to see whether or not this variant disrupts the function of the protein or not. How well a variant travels through a family. If you always see family where if there's the variant is present in homozygosity or in compound heterozygosity, it's always going to be seen with disease versus it's not. Information like this can really help somebody understand how well we think or how established the link between evidence and pathogenicity is there. And so what we use is the ACMG AMP guidelines, which was published in 2015 in the Richards et al. Paper and has continued to evolve. And we really keep to those guidelines as strictly as possible. We want this to be a conservative estimate. We don't want to tell anybody that we think your prevalence is this and then have to walk it back. So we keep to these very strict guidelines as much as possible. So this is a very well-established method. It's the gold standard within the community. If you hear anybody say they perform variant curation in the genetic space, they are using these rules or some derivative of these rules. But we also have that process of these loss of function variants that I was talking about that we pull from NOMAD. And we've realized that there needs to be a different type of curation looking at these because it's a slightly different question. The ACMG and AMP guidelines are really talking about variants that have been seen in individuals and are coming out of clinical laboratories. The loss of function variants that we're pulling out of NOMAD or other population databases are being pulled out of research data. And that's a slightly different thing. And there's some nuances that really need to be thought about when you're looking at these variants. The first and most important one are the main types of errors that can come out of looking at these loss of function variants. And the top one is really specific to research data. So research sequencing means that the difference between research sequencing and clinical sequencing is that if you have your genome sequenced in a clinical genetic testing lab, any variant they find and gets returned on the report, it has to be confirmed by a secondary method and has to be have two independent analyses that say, yes, this variant is in fact present in this individual. With research sequencing, we do a single pass and we say, okay, this was identified. And especially with NOMAD data, this was often for common disease, and we're looking at very large consortia. And so there's a certain level of error, but that's okay. That's baked into the research model. What we're looking at here, we really want to make sure that if we're counting a variant as being seen in an individual, we really feel that variant was present in that individual. And so we want to look for something called a technical error. This can be a sequencing or a reference error, but we want to make sure if we're including it, we feel confident it was really there. The next is a rescue error. As I mentioned, the high confidence loss of function is an annotation from a pipeline. What these pipelines can often miss is what's going on around that variant. Is there a second variant right next to that first loss of function variant that when you combine those variants together, because they're in the same codon, it actually leads to a missense, or even we can sometimes see that there's a splice rescue downstream from the variant. So the loss of function algorithm says this would disrupt splicing of the exon and intron. But when you look upstream, there's another splice junction only 3 base pairs down. And instead of loss of function, it's the insertion of a single amino acid, which isn't weighted as heavily. We can also see impact errors. That was not as much of an issue for the POLR3A and POLR3B gene, but we do see this with some genes where a variant is found to be loss of function, but it's in a part of a gene that's not relevant to the disease. So this can be last exon. This can be an exon that's actually not even present in the transcript that we're interested in for a particular disease. So this is all information that's really important to look at, because if we're pulling in these variants and counting them in the prevalence, we want to feel confident they're really leading to disease in an individual. And we can't always be 100% confident in that, but we want to rule out any major red flags, especially when we're pulling in these ones that haven't been reported in individuals before. So after we've performed that curation, now we're talking about specifically POLR3A and POLR3B. We've created the variant list. And so for POLR3A, we had 78 variants that had that ACMG curation performed, 82 that had loss of function. There was heavy overlap with how many got both. And so we had 126 variants used in the prevalence estimate. They really range the spectrum from 16 pathogenic, 65 likely pathogenic, meaning we're about 90% confident they're pathogenic. And then we had 45 that were uncertain significance. Now, for uncertain significance, we're not including all uncertain significance. These are variants that at some point somebody called them pathogenic, but when we put them through that gold standard and we're very conservative, they didn't quite meet the mark. Now, if one came out and we said, oh, this is because it's using very old information, in fact, we think it's more likely benign, we don't include it. We're really including ones where there is some evidence weighing towards this could be pathogenic or likely pathogenic, but in really rare disease where there's not a lot of patients and in places where functional studies are quite expensive and hard to find, we don't want to rule out these variants because they weren't able to yet gather the information they need. And so we include them, but again, we give sort of a range of here's the very conservative ones and here's a bit more relaxed. And I'll show you how those numbers kind of span out. For POLR3B, we had 38 variants that were ACMG curated, 75 that were loss of function. Little less overlap there where we had 94 variants used in the estimate, 6 pathogenic, 53 likely path, and 35 VUSs. So once we have that variant list, like I said, we now move on to the math. And this is the part I love the most. My variant curators do a ton of work and then they hand it off to me and I get to spend a lot of time in Excel sheets and kind of figuring out what's really going on here. So we start with first summing up all the allele frequencies for all the variants that we included. So there's a few things I always have to think about when we're summing up these allele frequencies. The first is we do it not just across the entire NOMAD database for the total allele frequencies, but we also do this for the 8 global populations. This is like non-Finnish European populations, African, Latino and admixed American population, East Asian, but also the subcontinental populations. To kind of give you a visual, these are populations that cluster within a smaller population. So for non-Finnish European, you can think about Bulgarian, Estonian, Northern European, Southern European. For East Asian, we're thinking about specifically Korean or other populations that weren't able to cluster into their own populations. But the key thing to remember is that not all these populations are the same size. And so there's a couple things we do when we're thinking about that. We remove any population that has less than 2,000 alleles, which means it's less than 2,000 individuals. The reason we do that is that when you have that small of a number, even a single change in your numerator, so 1 out of having one individual population can cause these very dramatic swings in allele frequencies and can sometimes make it look like there's a much higher allele frequency than another population, but they both have only been seen in one individual. And so we've really tried to say, you know, if it's less than 2,000 alleles or less than 1,000 individuals, we're really not comparing apples to apples, and we remove those populations from these calculations. But we still have some smaller populations that are only 3,000, 5,000, and other ones that are, you know, 100,000, 70,000. And so it's when I show you the graphs, it's just something to keep in mind that the smaller populations could have these like little spikes that make it look like it's a higher presence in a certain population. But if it's also a smaller population, we really have to be cautious about saying that's a true trend versus just a sort of an artifact of the smaller number of people that you're looking at. Again, you're probably sick of me already saying it, but it's something I mention at every pass, that not all populations are represented in NOMAD. And so if it is not seen in a certain population, or that the numbers are showing lower, or we're not seeing it at all, there are certain populations that are just not present in NOMAD at all, and therefore we're not able to assess their population or understand their contribution to the overall clinical population and spectrum. Final step is applying Hardy-Weinberg equations. So for those of you working in genetics, I hope you know what Hardy-Weinberg is, but for those who don't, Hardy-Weinberg is an algorithm that represents the theory that the amount of genetic variation in a population will remain constant from one generation to the next, and will have a predictable distribution of genotypes, which means if we look at one generation and their distribution, we can then apply it to think about future generations as well. There are a lot of assumptions that go along with that, assuming random mating, meaning any two people would come together and could have offspring. The other thing is that there aren't outside sort of bottlenecked effects or limitations on a certain population being able to mate with other populations. And we know that's just not true. We do know that there are populations that are isolated. There are higher levels of consanguinity in certain populations, but it does allow us at least a general approach, and our global sort of society is becoming more connected than it ever was before, which allows this to be leaned on a little more heavily. So you see the algorithm, which is a binomial equation up at the top. But the cool thing is for really rare disease, the P, which is the value that is expressed of the allele frequency of the more dominant, the wild type, the normal, the reference allele, if you will, is so close to 1 that you could actually assume it to be 1 in your calculation, and it makes the math quite simple. The math is that you sum all of the allele frequencies. So for conservative, we'd say likely path and path. We take all the allele frequencies for that variant. So numerator and denominator, we saw 1 out of 20,000 people for each of these variants. You add it up, and that is your aggregate allele frequency. Now, if you want your carrier frequency, you times it by 2. And that's really simple. We all get 2 alleles, 1 from our mom and 1 from our dad. And so we take the chance that we get an allele from somebody, and we times it by 2 because we get 2 pieces of information. For a disease frequency, you square Q. And so it's quite simple in the math, but again, it's the context and the nuance around it that can make this quite complicated. There are groups that perform this analysis with a little bit more of a Bayesian, which is a mathematical approach for weighting out sort of biases and things that we know about this. There's approaches for sort of trying to give confidence intervals, especially around the size of the populations. The reason that we've decided to keep this quite simple and not include that the Bayesian approach or the additional factors is because a lot of those introduce additional things that you need to think about and additional caveats. And we already know there's so many caveats with this. We want to keep the math simple so that we can really kind of assess it. And we find that actually giving more of this conservative to relaxed range, deciding to include certain variants versus not, gives you a better sense of what the range is than putting confidence intervals on every single data point. So we do return the results with all the allele frequencies and all the math that any group could then decide to apply the Bayesian model. We have a couple groups that they have a statistician or a researcher who applies the Bayesian models on top of what we do. We fully support that. It's just not what our group does. So these are the results. So we'll go through Polar 3A and Polar 3B and talk about what were some of the interesting trends for each of these, and then talk about them as a sum. So for Polar 3A, we found a conservative carrier frequency of about 1 out of 228, with a conservative prevalence of just a little bit above 1 in 200,000. So if we include those variants of uncertain significance that I talked about, it actually doesn't change dramatically. Those variants of uncertain significance didn't have very high, super high allele frequencies. So it kept it pretty tight, with a relaxed carrier frequency of about 1 out of 180, and a relaxed prevalence of about 1 out of 130,000. So at first pass, you look at this and say, wow, that's a really tight range. That's very simple. Here's sort of our range. Here's our answer. But it's when you start to dive into the variants and learn things that you actually can discover very interesting trends. And there's one particular variant that really jumped out in Polar 3A that changed the way that we looked at that range. And I'll get to that in 1 second, but I first want to just talk about Polar 3B's overall results. So for Polar 3B, we had a conservative carrier frequency about 1 in 526, with a conservative prevalence of 1 out of 1.1 million. The relaxed carrier frequency, however, when we included those variants of uncertain significance, was a huge jump to about 1 out of 200, and a relaxed prevalence of about 1 out of 180,000. So this is very different. This is a very dramatic change, and is saying, oh my goodness, there must be a ton of variants that we need to look into to really to figure out the prevalence. Well, when we dove in and started looking at the variants, so what are the variants that are contributing to this jump? There was, again, one variant that was really the one that stuck out. And so when we returned these results to the Yaya Foundation, both times we said, we know that we're giving you this range, but it's really one particular variant in both of these genes that changes the way that we think about these estimates. So going back to Polar 3A, this is now a different way to view the exact same table that I just showed you, but it allows you to kind of see the trends throughout the populations very, very easily. So again, we see just in all populations a carrier frequency of about 1 in 200. We see a little bit higher of a trend going on here. This is the non-Finnish European global population, followed by its subpopulations from Bulgarian all the way through Southern European and Swedish. And it is a little bit higher. We do see this, and it's a bit of a uncertainty. This is our largest population. We know in genetics that we are very saturated for the non-Finnish or the European populations in general. And so seeing a presence a little bit higher can sometimes feel like a self-fulfilling prophecy, and we're not always sure whether it's truly that it's just higher in that one population, or if it's actually, present in other populations as well. We just haven't found the variants that cause disease because that population is under-sequenced. But certainly, there does seem to be a higher prevalence, and we talked about that. But what was more interesting was that there was one variant when we were doing the variant curations that had a very high allele frequency and just kept coming up in a lot of reports. And this is the variant C.1909 plus 22 GA. And when we removed that variant from the math altogether, you can see the dramatic difference in how much these frequencies drop. Not only do we see a dramatic difference, we see a dramatic difference across the board. So this is not just isolated to the European population. If I click back and forth, you can also look at the African and Latino population, the Ashkenazi Jewish population, and you'll see this lower jump. So this is a global variant. We're seeing it across populations, and we're seeing it at high enough frequencies that without this, it really changes the overall prevalence. And in fact, it changes the prevalence enough that the carrier frequency drops to about 1 in 600, and the prevalence would drop to about 1 out of 1.5 million. Now, the interesting thing about this variant is we're not questioning its pathogenicity. The evidence is there. It causes disease. We see it in homozygosity in individuals with disease, but we're not really seeing it in 4H. So Polar 3A has a couple different phenotypes and likely represents a phenotypic spectrum, but we're not seeing it in the 4H leukodystrophy yet. in any of the case reports. It could be out there, but we haven't seen it in cases just yet. We're seeing it in the more severe WRS phenotype, and we're seeing it in ataxia. And even interestingly enough, in the one individual that's reported in homozygosity and ataxia, it wasn't even a later onset ataxia. It was actually a very early onset at age 11. But we're also seeing in the literature an individual who was only picked up because his child had WRS. He had carrier sequencing performed where they just looked at the variants that were identified in the child to figure out who had whether it was mom who contributed the variant versus dad. And dad was found to be homozygous for this variant. And he did not have disease that they could see at this point. So the question for this variant is not whether it's pathogenic, but how penetrant this variant is and what part of the spectrum it's contributing to. And that's really the lens that we were talking about. So we see this range, but it's the question of where in this range. And that really hinges heavily on what is the penetrance of this variant. Yes.
Can I ask a question?
Yeah.
Could you explain the molecular detail of this particular variant? What is 1909 plus 22? What does that mean? Sorry, I'm unfamiliar with the annotation.
It's a great question and actually plays into why we may be seeing this spectrum. So C.1909 plus 22, what this means is that 1909 is the splice junction. It's that boundary between the exon and the intron. And for those who are not familiar with genetics, it's the exons that come together to make the protein, and the introns are removed. So the sequence of the intron is not included in what makes the final protein. However, there's a lot of information specifically around the boundary of that exon and intron that is involved in the splicing process. So if you disrupt around that splice area, you can actually disrupt whether introns are kept in or whether an exon is skipped. And the plus 22 means that at position 22, we've seen a change. And around plus 22 is often a question mark, because this isn't really typically covered even in exon sequencing or in traditional gene sequencing. It's too far into the intron. Not all clinical labs cover this. Something covered more in genomes. But we know that these variants can actually impact things. And so it's not to say that if you see plus 22, it's not going to cause disease. It's just, it's sort of into a deeper region that it's more than likely impacting the splicing. The cool thing about splice variants, or not necessarily cool, but to somebody like me, what's super cool is that the mechanism is can be leaky, meaning that it's not every time that the variant disrupts splicing, and where your cells and where your DNA fixes it can actually change within a person. So you can say like 22 of your protein sequences skip this one or skip this exon. The other ones actually retain another one. And so it really leads to us thinking this supports the hypothesis that it's a spectrum, that some people have very severe disease, other people, their body may rescue it more appropriately and it doesn't impact it. And so it's not too off the mark to see this type of splice variant lead to sort of more of a variation in the way people present.
So I take it this one hasn't been characterized at the molecular level?
There have been some functional studies, but not, and Moriel Singerberg, who does a ton of this work,
okay,
and
thank you very much.
Into it, is on the call, so they can let me know.
Thank you very much.
Yeah, they, what I believe is we've done some functional studies where we see that it does impact the protein overall, but that like actually looking at the mRNA, looking at the protein itself and seeing exactly how many counts go which way, that can differ across patients. And so I don't think there's been a ton of work into that. And so one of my key things was talking to Valerie and to the IF Foundation about this is a cool variant that I think is an opportunity for them to look into and to try to better understand what's the spectrum. Is this playing a part in 4H? Is it just an ataxia and WRS variant? And it really changes the idea of how do we think about these gene disease spectrums, and from patient organization standpoints, where you draw the lines can be quite difficult. It's quite.
I agree. I think it's really interesting. Thank you very much for that very helpful explanation.
Oh, happy to help. Any other questions about this particular variant or anything I've kind of covered so far? I'll go into polar 3b. Great. So polar 3b also, as I mentioned, had one particular variant. And so we'll start with sort of the overall trends. We saw a carrier frequency of about 1 in 500 for all populations. This one was a little bit more, it wasn't quite as high. It kind of went across. We see this little spike in Bulgarian, but remember that the Bulgarian population is a little bit smaller. And so seeing, you know, even single individuals, we can see a little bit higher of a spike. So I usually try not to get too excited about this unless we're seeing one particular variant driving this higher frequency or a few that have repeatedly been seen quite a few times. But the interesting thing with this was the change between conservative and relaxed. So I'm going to change the color slightly on these slides because I'm going to change the scale. So this is the same information, but at a slightly different scale. So I'm just upping sort of my upper limit, so that I can show you the change between the conservative and the relaxed. So you can see it goes up quite a bit. And even in the Estonian population, but East Asian population, we're seeing this big jump. And so typically we could say, well, this is just a lot of variants and they have higher allele frequencies. So we got to figure this out. But when we went line by line and looked at the variants that were driving this increased frequency, I'll go back to sort of my original scale. There was one particular missense variant that really caused this huge or led to a big jump or contribution of the overall big jump. And so again, I'll just go back and forth so people can kind of appreciate. It was primarily in the European population. And the interesting thing with this variant is, again, there was evidence to support that it was pathogenic, but it met the VUS criteria. We really just couldn't get it there. It's a missense variant, which means it just changes one amino acid to another amino acid. These are variants that are harder to prove that they're overall pathogenic. We know historically missense variant in rare disease is just really hard to get there without functional work. But this was having a big contribution. In fact, changes the carrier frequency from closer to the 1 in 600 area to, or sorry, 1 in 500 to higher about, I think it was about 1 in 300 with this variant included. And so we really want to make sure that we know, you know, is this variant causing disease or not? Because the prevalence goes from 1 out of 1.1 million to 1 out of 400,000. And so figuring out this variant really will do two things. One, if it's pathogenic, we know that this is more prevalent than we originally thought. And here's, you know, again, kind of narrowing that window. If it's not pathogenic, we can really start clearing this up for a lot of patients that are getting this VUS on a report and don't know what it is. With a variant that is this high frequent, we know there are going to be people who have this variant that are out there that are being told, we think this is your clinical diagnosis, but you have one pathogenic and one variant of uncertain significance, and we're not sure. So we're just going to tell you this is likely your clinical diagnosis, or we're going to leave you on the hook. And so being able to start looking at these types of questions of saying, where are the places that we should put our resources? Functional studies are incredibly expensive. They're hard to put together. We know that in genetics. Now we can start to look at, let's look at the allele frequencies and the contribution of the variant to the overall spectrum to decide how to tell patient organizations or researchers to spend their money best. Where's going to have the highest impact? Not that we by any means should ignore other variants, but we have to think fiscally and as a budget and as a group of where are we going to have the most impact. And this is a variant that I think we can just say very quickly, here's the evidence to say this is a variant that we need to figure out. So then moving on to just putting these two together, we know that it's polar 3 as a unit. A and B on their own is not necessarily as informative. So this is a slide putting together the two. The conservative carrier frequency is about 1 out of 160. And you can see sort of the contribution of the two genes, with B being the darker and A being the lighter purple. And then the relaxed frequency jumps up quite a bit. Again, this includes that missense variant that I was just talking about. So you see it go up quite a bit. Now with sort of the relaxed carrier frequency, we have a closer to about 1 out of 110 carrier frequency combined. And our conservative prevalence, it's hard to appreciate this because I wanted to have the scale so you could see the jump with the relaxed, but it means that I kind of minimize the amount you can see in the other smaller populations. But we have a conservative disease prevalence of about 1 out of 175,000 individuals, and a conservative of about 1 out of 175,000, and relaxed about 1 out of 75,000 individuals. This is math that I always tell everybody not to do, but if you're going to do it while I'm talking to you so I can talk to you about all the caveats, but it's very natural and very human to take the overall prevalence that we give everybody and they times it by the world or the US population, and then they say this is the number of individuals. This is riddled with caveats because NOMAD is not representative of the entire world, and we know that there's a lot of biases built into this, but it's also very human. I think we take numbers and fractions and we want to times it and put it into context of individuals and people, because that's something we can understand. So while I know that it's not the right thing to do mathematically, I also know absolutely everybody that I talk to and return these numbers to do this. So I want to give these numbers. With these numbers, I just want to say that the Polar 3A, we are including that +22 variant in these numbers. And with the Polar 3B, these are the numbers without the missense variant. So these were sort of the conservative numbers that we gave everybody, but just remember those ranges depend on whether or not you include these two variants. So coming back to my initial spectrum. So there have been publications for Polar 3. We are not the first people to do this. There's publications talking about the clinical cases and the incidence and the prevalence, but we know that that's likely undercounting. There have also been publications that did this method that we did before, but they didn't include the clinical curation. And so they had closer to our relaxed numbers, if not a little higher, because they also included some of those loss of function variants that we looked at that we ended up ruling out and saying there's reasons to say that these are not loss of function. And so we feel pretty confident that when you look at these numbers and that when we're kind of looking at the spectrum, that we think we're here. If you include that +22 variant, knowing that not everybody with that variant likely or homozygous for that variant leads to disease, that it's such a frequent variant, it's likely kind of overcutting it just a hair. But we also know that Polar 3B potentially is a little higher, but that is really slightly a different question where we're saying a binary of whether we're including or not. So my gut is that really we might be, when we're looking at those numbers with the +22 in there, we could be just a little high above it, but that we're getting closer and closer to the true genetic prevalence. So again, talking about these two variants and why I think that, the +22 is really, again, it's a range, and that the true genetic prevalence is somewhere in between there. And that really depends on us understanding the penetrance of that +22 variant. With the Polar 3B and the missense variant, that's a binary question, whether we include that missense variant or not. So either that variant is benign and the prevalence is closer to 1 out of 1.1 million, or it's pathogenic and the prevalence is closer to 1 out of 400,000. And so this is the really cool thing and the cool story about looking at these variants is if we accept the variants at face value and those numbers at face value, again, Polar 3A looks like it's this really tight range and Polar 3B looks like this giant range. But when we start to dive in, the story clears up a little bit more. And in fact, we can understand that there's different types of questions where we're just shortening a range or we're actually moving these benchmarks and we're saying it's either here or there. So coming back to my puzzle pieces, pulling away from Polar 3, although I think Polar 3 has helped us appreciate a lot of these, we've done this with a lot of different groups and we've had some great lessons learned. So we started with this puzzle. These are the things that we knew. These were the tools and mechanisms we knew how to apply. We felt very confident in it. But as we've done this study now over 18 different genes with different patient organizations, there's additional things that have been learned. One, I think is the most important, is that partnership with these patient advocacy groups is an absolutely critical piece of this. They have access to the researchers, they have access to patients, they can understand the natural history. It is not often that, or not infrequent, that we say this is a key variant that we need to know, and they can very quickly work with their organizations, work with their researchers to figure out how often are we seeing this variant? Do our researchers know anything? Can we get access to functional studies. They're a key source of information and are absolutely imperative collaborators as we go through this process. As we move into classification, there's two things that we need to think about. One is gene novelty. I talked about those 3 positions that we could be on the spectrum. Sometimes there's a fourth that's actually off my screen, which is we don't know the true variant spectrum. And even doing this method, we are undercutting it because we don't know all the variants. Those are usually much newer genes that we don't know the full sequencing yet. And you need to think about that. So gene novelty and how well that gene disease relationship has been established is absolutely something you need to think about when you apply this method. The other is functional work. That is really a key piece to trying to figure out this variant list and knowing which variants to include or not. We have partnered with groups that have a disease assay specific to their disease, and it makes this work so much faster and so much easier because we can just look at the assay and say, did this variant meet the criteria or not? There's still things to think about, but it's really a key piece. But we know it's expensive, we know it's hard, and we want to partner with these groups to say, here are the key things to go look up. When we're talking about aggregate frequency, I can't say it enough, representation. But looking at this not just as a downside, but also an opportunity. This is now math and algorithms and proof of concept that by not having full representation of populations in databases is hindering our ability to help those patients and to help diseases in general. And so as the operations manager for Nomad, we know this and we are moving towards, helping to increase the diversity of Nomad. We are now months out from launching version 4 of Nomad, which will have over 800,000 individuals with far more diversity than past things, but moving towards our next versions of Nomad, including all of us data, and including the diversity, and also working to a federated data model where we're able to have people use the infrastructure of Nomad across the world so that protected populations where their data can't leave their boundaries actually could set up their own instance of Nomad, and we could share allele frequencies so those people can be protected. They can have the resources go to their country to set up these databases and to have the regulations they choose to have, but that we can still aggregate that information together and improve representation for everybody. And the last and key thing is to always think about your disease, the disease spectrum. Do carriers have disease, have phenotype or not? Is it one disease? Is it multiple diseases? It's absolutely critical that we think about this and put that into the context of the prevalence discussion. All of these are data points that really need to be brought together. None of these can live in a silo if we want them to be truly impactful. There are limitations to this method, and I'm always very honest about them. We only include variants that are present in Nomad. This is a hindrance, but again, adding 0 doesn't really add anything. We know there's pathogenic and likely pathogenic variants not found in Nomad. Not all variants of uncertain significance are included. So if there are missense variants that we don't know much about, unfortunately, those aren't included. We know some of these missense variants at the molecular level lead to loss of function, but we need to wait for that evidence before we can include them. I think I've mentioned enough times that not all populations are present in Nomad. And again, this calculation assumes random mating and doesn't take into account increased rates of some consanguinity in some cultures and populations. And then the last thing I want to leave off on is that this is by no means something that we want to keep separate to ourselves. And in fact, are working very hard. After version 4 of Nomad is coming out, we will be releasing a calculator that allows anybody in the world to go online and perform these calculations themselves. Which is why I want to train everybody about the caveats and the things that they need to be thinking about. But with those tools and with being able to think about this context, it will allow patient organizations and researchers to perform these methods and be able to get out these results quickly and into the hands of patients and patient organizations who absolutely need to have this information. You'll be able to print out final reports that can be shared with other interested parties. And we may even move to the point that you'll be allowed to make your variant list public. That will likely be a later version, but that you could then just have a URL that you can say, here's my variant list, here's the math that we applied, go look here. And I think it will even expedite the process even faster. So once again, I am by no means a single person doing all this work. It is a large team of people doing the variant curation, but also working on Nomad and the methods that we apply. And so I am representing an absolute village of our translational genomics group and rare genomes project. And thank you to the Yaya Foundation and CZI for partnering with us. We could not have done it without them. With that, I'll stop sharing and see if there's any other questions.
Well, thank you, Sam. That was a wonderful talk, and I'm glad that you could show, that we're actually not as rare as we thought, and
definitely not.
Which, many of us, I know I was one of the first people, one of the first things that I did after you sent us the carrier frequencies was exactly that, what you said, we shouldn't calculate the populations. But it is, no matter how we look at these and how much we say, well, maybe it's only half,
Yep.
How far we go to the conservative part. There are more patients in the US or in the world, but for us right now, than we know of. And it just emphasizes that we need to go to look for those. But anyway, aside from that, I would love to hear questions. If you have questions, please turn on your video, but any questions out there?
Hey, Samantha.
Hi.
I have a question. So the data that was presented include prevalence from mutations in 3A and 3B. I was wondering if you had data on mutations in different subunits that also cause polyp leukodystrophy.
So we, you mean different genes that cause the leukodystrophy?
No, mutations in different subunits. So I'm thinking of A.
And K.
Sorry.
POLR1C and POLR3K, I think, are the other genes. We did not submit those because they are so far so exceedingly rare. That's the caveat that we should add. We have not all the genes included, but we decided in the interest of time, because it is so much work to curate those, to leave these off by now, but it's a logical next step. Thank you.
And it is a limitation of just like the funding. So we were working with all the Rare as One advocacy groups, and we had 2 years to perform this. So we limited each group to 2 genes. So that was baked into sort of the model, but that is the point of getting the calculator out there. And it will be available to anybody. And so we are trying to work with groups to have them understand how we perform this method so that they can apply it to any gene that they need to. So part of this is Valerie and the whole foundation has access to us. And so if there are other genes they already know about, we can help them kind of set up those lists, think about it, and then the list will be able to be shared across groups. So like Valerie could set it up, or you guys could set it up and you can share with each other. You'll be able to add notes to the list, check and uncheck a variant and say, let's include this, let's not include this. And then we will notify you if there's new versions of Nomad. So you may want to redo it with new allele frequencies, add additional variants as they're learned. That's incredibly important, especially for newer genes where variants are still being found, especially for really rare ones. Adding every single variant counts. And so the tool will help facilitate with all of that.
All right, great, thank you. I have another question. One of your final slides, you had a prevalence number. It's 1 in approximately 4,800. I'm wondering, that number is the combination of the 3A and 3B data that you presented. Is that correct?
Sorry, say the number one more time, the 4,300?
No, 1 in 4,814. That was the global prevalence that you had on one of your final slides. So
yeah, so that 1 out of 4,8 was if POLR3B, if that missense variant is causing disease, the overall conservative prevalence would go up to about 1 out of 400,000. The final conservative prevalence of the two combined was 1 out of 174,000. 1 out of 175,000 is probably the more appropriate one.
Okay.
Yep.
And one final question.
Yep.
You mentioned that 3A prevalent variant was not classified as 4H. It was classified as WRS or ataxia and was identified based on population genetics. Can you elaborate on that, please?
So this variant was, as you guys probably know, 3A has a spectrum. When we do this method, we do it at the gene level because we're saying, let's bring in all the variants for the gene. Because we knew about this variant early on, or we knew that this gene had a spectrum early on, we annotated each gene to say, is it seen in one of those three, WRS, 4H, or ataxia. This, the plus 22 variant in the literature that we looked at to date hadn't been reported in somebody with 4H. It was only in individuals with WRS or individuals with ataxia. So what I was saying is it hasn't yet is because there isn't, that relies on people going out and doing testing. There wasn't a comprehensive review of every single person who has been reported with 4H to know whether that variant. So at least from our standpoint as a curation team handing this off, we thought that that was a question to ask, is does this variant contribute to the 4H prevalence versus is it just WRS and ataxia?
Got it. Thank you.
Yep.
If I can just add something, or maybe somebody from the McGill Lab has to add something to that. I think I recall a publication that had that variant, the splice variant, and hypodontia. So it seems like, once again, it is the issue was the spectrum, and we didn't just want to cut off, a whole part of the sort of milder spectrum. That's why we decided to include that.
So
have it in some of the calculations, but it is true. I mean, it is when you take it out, it's the issue with the spectrum. So, yeah. Somebody had a question, sorry.
I was gonna say, if you're referring to the 1909 plus 22 variant.
Yep.
It's in the chat. There's someone put it in the chat.
Yeah.
A link to a paper.
Yeah, Moriel did.
Yeah, sorry. I just put in a link, one of the functional characterizations of that, since I think there was a question on that earlier, but there's like plenty of publications on that variant if you want to take a deeper dive.
Are there any other questions? And like I said, I can stay on so people there were a lot of ways that you can parse this data and look at it. Give it only an hour. I tried to kind of hit the major points, but if there's anything else people want to ask questions on, I'm happy to stay on and talk.
Well, if not, as I said, this is recorded and we will put this up on our channel, so we have an opportunity to review it. And please, if anybody has any further things or questions, please contact us. And thank you, Sam. Thank you for giving this presentation. And I'll see you hopefully all soon at the next seminar from the Yaya Foundation.